Hello students, today we will see biomedical waste. So its definition, rules of 2016, the types of biomedical waste and its hazards associated with the biomedical waste. So according to Biomedical Waste Rule 2016 of India, biomedical waste defined as waste that is generated during laboratory diagnosis or treatment or immunization of human being or animal, or in research activities or in production of biologicals. So that is called biomedical waste according to the rules of 2016 of India. Now, aim of waste management to prevent harm resulting from waste, to minimize its volume, to retrieve reusable materials from the waste, then to ensure safe and economical disposal methods, and last to protect health and well-being of patients, staff and public. So what are the steps for this waste management? So first is the reduction. So proper planning so that the waste volume should not increase. So main first step is the reduction of the biomedical waste. Then after that, whatever waste is covered, so you have to recover that. So reuse. So proper planning of the reuse of the waste produced. Then segregation. So at the point of generation, the waste should be segregated into different color coded bags. Then proper storage, then transportation to the central unit, then treatment, that is procedures and process to reduce bulk of waste and make it non-infectious and harmless. So these are the steps of waste management. So what is waste hierarchy? So in that all R are there. So prevent and reduce, that is production of waste is prevented or reduced to minimize quantity of waste. Then reuse and recycle. So plastic material should be recycled. So whatever reuse and recycle possible from the waste produced, that we should allow. Then recover. So energy recovery, that is waste is converted to fuel for electricity, or composting of organic waste to produce compost used in agriculture. So that is recovery of the waste. So to make it useful from the non-useful waste. Then treat. So whatever now leftover waste that cannot be reduced, cannot be reused, cannot be recovered, should be treated properly to prevent health hazards. So the treatment example like incineration or autoclave, and then disposal. So that should be disposed properly in landfill or dump yard, so that public or society should not get harm from that waste. So the waste are divided into two types, non-hazardous waste and hazardous waste. So non-hazardous is harmless waste, that is general public waste, that is kitchen waste, or we can see the waste from the leaves or plants. Then hazardous waste is infectious and non-infectious. So infectious waste is biomedical waste, that is hospital waste, and non-infectious hazardous waste may be chemical, radioactive, pharmacological or cytotoxic. So now we will see according to the WHO classification there are few categories. So first is general waste. So it is no risk to human health. Example, office paper, wrapper, kitchen waste, general sweeping waste. Then pathological waste. So whatever waste generated in the pathological laboratory like blood and other samples, body fluids. Then sharps. So it can be needles, scalpel, knives, blades. Then infectious waste, which may transmit bacterial, viral, parasitic diseases to human beings, and waste suspected to contain pathogen. Example, laboratory culture, tissues, then bandages, dressings if they are infected. Then chemical waste, so it is laboratory reagent or disinfectants used. Then radioactive waste, it is unused liquid from radiotherapy or lab research, contaminated glassware while doing investigations. So this chart will show you the category and the treatment method. So biomedical waste management, first is segregation of waste, then collection of biomedical waste, then transportation to the central unit, then treatment of the hospital waste. For the solid waste it can be incineration or autoclaving. For liquid waste it can be disinfection. So human or animal waste or soiled waste can give infections like HIV, hepatitis B, hepatitis C, hemorrhagic fevers, cholera, salmonella infection, shigella infection, then rabies, leptospirosis, anthrax, tuberculosis, pneumonia, septicemia. So these are all associated with the human or animal waste or infected waste. Then sharps can transmit infections like HIV, hepatitis B and hepatitis C. Then cytotoxic or radioactive waste can cause cancer, genetic mutation or birth defects. Then chemical waste can cause poisoning, dermatitis, conjunctivitis, bronchitis. So there may be health hazards like psychological stress, infectious or diseases or injuries, cancer, genetic mutation, birth defects, poisoning. So all these are associated with the hazards of the. So what are the different types of waste generated in the hospital? So they are solid waste, liquid waste, biomedical waste, chemical waste, radioactive waste. So different types of waste generated are these. So we will see in detail. So it can be category 1, that is human anatomical waste like blood and blood products, placenta, removed tumors, other surgically removed body parts like appendix, uterus or biopsy materials, excreta. Then category 2, it is animal waste. Category 3, microbiology and biotechnical waste like culture media and plates, vaccine waste, reagents used in microbiological laboratory studies. Then category 4, it is sharp waste, that is needles, syringes, scalpels, glass beads, broken ampules. Then category 5 is discarded medicine and cytotoxic drugs. Then category 6, soiled waste like cotton dressings, plaster cast, soiled beddings, linen, clothes, mask. Then category 7 is solid waste like plastic waste like IV sets, tubings, catheters. Category 8, that is liquid waste, which are reagents used in the hospital. Then category 9, it is an incineration incineration ash. And then category 10 is chemicals. So the yellow bag, it is a non-chlorinated plastic bag, and separate collection system leading to effluent treatment system should be there for the chemical liquid waste. So this yellow bag contains human anatomical waste, expired discarded medicines, then chemical waste, microbiology and biotechnical clinical lab waste material. And treatment or disposal method is the incineration, plasma pyrolysis or deep burial. Then red color bag, it is non-chlorinated plastic bag or container, and here mainly it is recyclable contaminated material is there like tubings, bottles, IV sets, tubes, catheters, urine bags, syringes without needles and glues. So the treatment method or disposal method, it can be autoclave, microwave, hydro processing or recycling and then to the landfill. Then white color translucent puncture leak proof container. So mainly it contains waste sharps including metals, and then method of the disposal is autoclave or dry heat sterilization, then shredding of that or mutilation, and then encapsulation for the reuse. Then blue color contains, it is a cardboard box with blue colored marking. So type of waste, mainly it is broken glass waste, slides, cover slips or pipettes or ampules without medicines. And then treatment, it can be disinfection, autoclave, microwave, and then recycling should be done. So there are steps we already saw. So first is segregation, then transport to site of temporary storage, and then final disposal. So segregation, the staff is provided with personal protective equipments. Then segregation is done at source where the waste is generated. Then color coded waste bags are placed in bins in all patient care areas. Labeling of all the bags with pre-designated labels with information including hospital name, patient care, unit name and date is done before use of bags. So transportation, waste from various patient care areas is removed twice a day or more frequently if necessary. All bags are tied at the mouth to avoid spillage during transport. Avoid the transport of too many bags at one time. Avoid contact of the bag with the body of personnel. Mixing of segregated waste should never be done, and biomedical waste bags should be sent to disposal when it is 3/4 fill. So these are all the points to be considered while transportation of the waste. Now disposal methods. So first is chemical disposal, chemical disinfection. So for sputum, pus samples or body fluids, blood, urine, stool, all they are done by chemical disinfection. So 1 to 2% sodium hypochlorite solution is used for the chemical disinfection. Then deep burial. So material after disinfection put into deep trenches covered with lime and filled with soil. So this is mainly done for shafts. Incineration. Solid infectious waste, anatomical waste under high heat converted into ash. So incineration is done by using high heat, that is burning, and then it is converted into ash. The disadvantage of this incineration is for the plastic, because of the plastic they under high heat they give form black smoke. So that is carbon monoxide production, and that is injurious to the health. So plastic items should not be incinerated. Then autoclaving can be done for infectious waste. Then microwave for small volume infectious waste. Then ETP, effluent treatment plant. So liquid waste treated with disinfectant. So liquid waste which is generated in the hospital, it is treated with the disinfectant, and then neutralization is done and then flushed into the sewer system. So that is ETP, effluent treatment plant. And then mechanical pressure shredder. So all glass waste or plastic waste should be mechanically broken down into small pieces, and then they can be reused. So liquid waste pre-treatment should be done 1 to 2% sodium hypochlorite used, and then ETP plant to remove suspended solids and organic matter, and then drainage system of hospital should be connected with ETP and outlet of ETT connected with the municipal drainage. So this is these are the disposal methods. Solid waste management. So composting by microorganisms and vermicomposting by earthworms, or landfilling for disposal of non-biodegradable waste. Sanitary landfill, open backyard, it is not acceptable, and recycling can be done. So biodegradable waste degraded by microorganism into simple compost, example paper or food material. Non-biodegradable waste non-degraded by microorganism, example plastic, aluminum cans, so they are recyclable. Now advantages and disadvantages. So landfill is a cheap, land can be reused once the site is full, can be used to deal with large volumes of waste. But the disadvantage, toxic chemicals can be produced, heavy metal ions can leak into the ground, non-biodegradable plastics may not be broken down, and it occupies large area. So that is a disadvantage of the landfill. In special situations like metal sharp, so needle cutter or burner for cutting or burning needles can be used, like blades, needles, scalpels. They're placed in white container and then it is sent to the central unit. Then glassware, pre-treatment with 1% sodium hypochlorite for 20 minutes, and then broken slides, test tubes stored in blue color puncture proof container. Vials, ampoules can be used put in a blue color puncture proof container.
Hope you find this session useful for your understanding and for your exams. Thank you.